and so I don't want to finish. There we go.
Good. Okay, there we go. Okay. Yeah, just to back up a little bit. So yeah, I've been doing this for over 30 years since I was a graduate student. And back in 1992, I was a graduate student looking for a thesis project, and I found one. And yeah, 1992 was a good year, just to put all the challenges in perspective here, and also the controversies as well. This was the year that that the first bold fMRI papers came out. The year before was the year that Jack Beliveau produced his landmark study using gadolinium contrast. And as portrayed here on this Science article, this was 1991. This was November 91. And by then, all of us were very busy, at least Ken and his group, and Seju Gao and his group, and myself, I came in a little bit later. And then a couple of other groups were busy as well. Andrew Blamer at Yale and Jens Fromm in Germany. So those are the first 5 papers published in 1992 on functional MRI. And without getting into too much history, but just to give you a perspective of what 1992 was like, this is just some things that might jog some of our memories, but some of you maybe weren't born then. I did all my analysis on a Sun Spark Station 2. And by then, just the first PowerBooks were coming out. All of our data, believe it or not, and this seems really old, but GE systems are always a few years behind in terms of their data architecture and things like that. I saved all my data on reel-to-reel tapes, 20 megabytes. So that changed within a few years, but then, you know, we did our word processing on something like a Mac Centris or something like that. So anyway, this is 1992. And back at the Medical College of Wisconsin, we actually just used the standard 1.5 Tesla scanner. We used a local head gradient coil to be able to, it had low inductance, so it was able to use the standard gradient amplifiers to drive it a little bit more a little bit faster. And we had different versions of this. And what's interesting is that, you know, things were a little bit more fast and loose then. We scanned in the middle of the night. We just stuck this coil on the table, not worrying about like if there was a short, it would torque out or anything like that. And we had to, you know, hook it up to the scanner and get it running. So that was 1992. And oops. And this is a movie, and I like to show this movie. This is actually one of our first successful data sets. This was back in 1991. This is my brain tapping my left and right fingers. And you can see the movie. It's a time series of difference images. But when you look at this movie, you see that literally everything that we're still struggling with, a lot of the things that we're still struggling with are all there. I mean, there's physiologic noise. There's some interesting other networked activity. There's, you know, pulsations, but then, you know, there's this slow, sluggish response that in the left and right motor cortex when you're tapping left versus right hand. So it's all there, and we're still working on that. And this is the hemodynamic responses that were induced either by a very brief stimulus or, you know, a long stimulus, and there's sometimes a post-undershoot. So just to quickly introduce the challenges. So after 30 years, what has improved after 30 years, after 30 plus years? So certainly image quality, temporal stability, sensitivity, resolution, and speed have improved. There's, you know, parallel acquisition that was a sort of a large improvement in fMRI. The implementation and also the standardization of the acquisition, processing, and display have improved as well. More groups are doing it. There's more standards. There's an explosion of processing methods that are much better than, or much more robust and adaptable and sensitive potentially than just simply univariate analysis. You know, there's decoding and coding, pattern effect mapping, or dissimilarity analysis, machine learning, cross-subject correlations, dynamic resting state connectivity analysis. They've all opened up new insights and directions. The artifacts are understood and more fully removed. The neurocorrelates of fMRI are better established. There's much more confidence that it does reflect neural activity consistently. Resting state fMRI, back in '96, it took off, but it took about a decade for it to really catch on. By 2006, it's exploded. Big data is a major improvement in the sense that we have these large pooled data sets that have catalyzed meta-analysis, more transparency, even new method pooling and reproducibility in biomarker discovery. Even though we haven't actually discovered biomarkers yet, it's catalyzing it, hopefully. Individual assessment in fMRI is growing. And I think this is actually, I'll talk a little bit more about this later. I think that's a big thing, which we're just getting our heads around. And that's part of the reason why our big data analysis isn't working, is probably because there's more individual differences than we fully appreciate. And to get our heads around that, I think will allow us to go forward. Multimodal integration, different techniques complement each other. There's more groups doing it. That always helps. That helps the field ratchet forward faster, as opposed to 1.5 Tesla, 3 Tesla is standard. There's about 107 Tesla scanners in the world as well, it's becoming standard. I don't know how sustainable it is because there's no actual clinical applications yet that are billable necessarily with 7 Tesla, but hopefully that will get there. So what has not improved? Well, we're still struggling. Since day one, we're struggling with subject motion and breathing. We still haven't quite solved that. You would think we would have, but we haven't. The temporal signal noise is still limited by physiologic fluctuations. We're still struggling with understanding how the hemodynamic response functions varies over space and across subjects. And we don't know how to normalize it in a robust way. Vendors, and actually this is an important point, is that vendors really drive most of even the fMRI technology, even though they don't really care about fMRI. We sort of piggyback on their systems, which has actually allowed fMRI to spread, but at the same time, they haven't focused on fMRI because there's no clear clinical applications yet. There's no clear, robust clinical applications. So they haven't spent much time trying to develop and optimize it. Scanners are still loud and confining. MRI is, of course, still expensive, which also is another impediment to making it more clinically applicable for fMRI. Many studies are still using 2.5 millimeter voxels, which was about maybe a little bit higher resolution than in the day one. We don't really understand, and this is sort of my opinion, but I think it's true, connectivity as inferred by correlation. When we see correlations, we say their areas are connected, but we don't really know that. There's a lot of things that can modulate connectivity that have nothing to do with correlation. That modulate connectivity that have nothing to do with connectivity. They can modulate correlation and have nothing to do with connectivity. And we don't really fully understand resting state fluctuations. We don't know their purpose. We don't know how much is conscious, how much is unconscious. And yeah, we still don't have our heads around that yet. Shimming. You'd think that shimming would be a solve, but especially at 7 Tesla, a lot of groups still, even at very high resolution, have a hard time getting temporal pole and subcortical areas that shimming is still an issue. There's still a limit, and this is a real limit, but there are ways around it or through it of the maximum rate at which you can switch gradients. To go to high resolution, you need to switch your gradients rapidly. Eventually, this causes peripheral nerve stimulation, and that's a real hard limit in many cases. Large databases, even though we have large databases, they're really not curated well enough. There's not any standards necessarily for the pulse sequences or the data format. There's a BIDS data format, and there's other types of data formats, but they're still in the early stages of evolution. And it's really hard because it's like hitting a moving target. The methods are developing rapidly. The pulse sequences are still developing rapidly. So it's hard to make these big databases with one standard technique. So fMRI, of course, as I mentioned, it's not a clinical tool yet. I mean, yes, it's used for pre-surgical mapping. And yes, there's some, especially at MGH, there's some amazing work on clinical applications. But as a tool, it's definitely not something that's distributed. Okay, so that's the field in sort of a nutshell from the way I see it. I wrote this book. It's a short book. It's called fMRI. This is what Bob sort of discovered the other day, and it has one chapter in it, chapter 8, that was a lot of fun to write. I just decided to expand on a blog post of mine called 26 Controversies and Challenges, and I list them all here. Initially I thought, oh, I'm just going to go through this whole list, and I thought that really wasn't. It would be fun for me, but it may not be, fun for everyone else to go through the list. So I decided to try to curate this list and sort of package it in different ways. And so I decided to first start with just talking about the controversies, because I don't really want to dwell on the controversies. I want to dwell on the challenges a little bit more, but just to categorize the controversies. There's like pretty much 4 types that I would call types of controversies. One is aspects of what we're doing that we might be doing wrong. Like for instance, a controversy was the use of global signal regression. It works very well for cleaning up the data, but it might, and I'll talk a little bit about this, it might potentially warp some of your results such that you see negative correlations where none exist. That's an example. And there was some discussion about that, and there's still no clear agreement as to what is best to do. It depends on the question and how you do your analysis. As I mentioned, connectivity change or difference interpretation. I really think we should stick to calling it correlation because, for instance, you might have 2 areas that have a high correlation, and let's say one area, the amplitude goes up. Suddenly, they look like they have a higher correlation just because it's a little bit more above the noise. It's not necessarily any more connected. Or you might have one area where there's a lot of pulsations, or the pulsations change. That would cause a change in connectivity. It doesn't mean those areas are less connected. So it's important to keep that in mind. Multi-subject analysis, how to pool the data. This came up, I believe it became more clear in this paper by Merrick et al. A few months ago, talking about the need for thousands of subjects to get any sort of reliable biomarker sort of signal. And I think it has a lot to do with how we're not embracing and adapting to individual subject differences and all the dimensions of how they could be different. So doing multi-subject analysis is pooling templates, like a single template parcellation, and then using that as the template might be throwing out a lot of sensitivity. And that goes into parcellation templates. The simple use of thresholding. Because we don't really know the nature of the noise, there's a lot of potential misinterpretation with thresholding. And this sort of links to what I'll mention here, this multi-center, this discoveries of shortcomings that get amplified by non-experts. There was a recent paper on where 90 groups analyzed a single data set. They got different findings, a lot of different findings. And I believe that was some of it of a straw man, because they actually used a pretty stringent, a lot of them used thresholds, and then they compared the spatial alignment after they used thresholding. And of course they were different, but there's a lot of real activity below threshold that we still haven't figured out how to set that threshold. There's no one statistical test that tells you how to threshold. And that's an important point. Of course, simple regressor models might miss a lot of information as well. So findings that are intriguing but have not been widely reproduced. Of course, there's been in the literature many studies on activation-induced changes in diffusion, neural currents, proton density, things like that, that have not been reproducible, that people still stick by them. And usually they sort of become marginalized, especially if they're not robust. Activation-induced bold and white matter. It's funny, the physicists actually argue over these things, and we love to talk about this because it's an interesting point. Nobody, people would like to see activation of white matter. Some people claim they've seen it. Some people claim they haven't. And so it's still up in the air. The pre-undershoot was something that came up years ago as potentially being more indicative of neural activity at more precisely located where neuroactivity was, where it was pulling oxygen from the blood before the hemodynamic response kicked in. I looked for it for a long time, or for a reasonable amount of time, and I never found it. And so now the field has sort of gone on. You can reproduce a pre-undershoot if you massively lower the blood pressure. So hemodynamic activation, but no neuronal activation. There was one paper by Curated and Das that claimed that they saw hemodynamic changes with no corresponding neural activation. I believe, and I think the consensus has since then been that there actually probably was neural activation. They just weren't sensitive enough to see it. And this one is truly hard to believe if it's true. And of course, related to the Merrick paper, there's a lot of small N group comparisons that are just not replicable. And this has formed what's called sort of a replicability crisis in the field that I believe Russ Poldrack and others are trying to head off and deal with a little bit more with larger data sets, more open data. So discoveries or shortcomings that get amplified by non-experts. These are interesting because these are all things that all the experts in the field just read the paper or saw the abstract or whatever and said, yeah, okay, and next thing, whereas the popular press picked it up or non-experts picked it up and said, oh my gosh, fMRI is horrible. So, the first one I think was the dead fish one, which was a simple study of false positives if you don't do Bonferroni correction. There was the controversy of double dipping, where you selected a region of interest and then you looked at the correlations within that region of interest. It was sort of circular. That was corrected. All these are sort of self-corrections of the field. There's a clustering conundrum where that algorithm for determining cluster size was potentially a little bit too generous. It didn't really make a difference for most studies, but the literature certainly picked this up and said, oh, well, maybe 20,000 papers are wrong because of this. It's like, no, they're right, but it's just a little bit slightly larger area of activation. And I heard he talked about the multi-center data set. I actually reviewed this and I warned the authors. I said, this is going to be picked up by popular press and they're going to say that, fMRI is all over the place, and which it was to some degree. And it's actually an interesting problem. It sort of shows, right now in this course, you're learning analysis pipelines. And you'll go back to your group and you'll analyze data and you'll produce results. But the fact is that different centers produce, they have very similar sort of pipelines that might vary a little bit, maybe in pre-processing or how they choose the thresholds or the regressors. And it will come up with results that can be amplified as different depending on how they choose their thresholds. So this is an important point. I think that there should be a reanalysis of this, a meta-analysis of this data to show how it might be very similar if you just tweak a few things in terms of the final results. And then the thousands of subjects, the big data conundrum. Basically, I believe that was a really wonderful paper that was really important to see, but I think it sort of highlights what we need to do to make biomarkers. We actually really have to get our heads around all the details of how you average subjects together. So there's a lot of mysteries. As I mentioned, what's going on with resting state activity? What neural activity does BOLD actually reflect? How relevant or informative is this spatial temporal scale of fMRI for actually understanding the brain? A lot of single cell electrophysiologists will say, well, we're at the wrong scale. We're just looking at this sort of epiphenomena. What is the global signal? So we're trying to regress it out, but it might be meaningful. And what actually is it? Is it breathing? Is it something else? Is it something neuronal? And along those lines, a mystery for myself is that global respiration regions, regions that that show up as showing correlated with respiration, they overlap with some resting state networks. So there's a lot of similarities. Jean Chen wrote a nice paper on this, and Rasmus Byrne, when he was a postdoc with me and a staff scientist, showed this as well. And so it's a little bit concerning that some of the strongest resting state networks seem to overlap significantly with respiration. All right, so that's the controversies. I'm going to go into the challenges now, and just starting by talking about what I like to put as 4 pillars of fMRI development. And all these pillars are basically things that where there's an advance in one, it usually helps all the others. And where there's an advance in others, it helps, they all sort of bootstrap each other up. They all open each other up. And some of the best fMRI work, some of the most intriguing findings, the most innovative work is right at the center of all these. It uses the most advanced methods. It interprets the signal as deeply as possible. And there's some interesting application that it's applied to. So what I mean by pulse sequences, of course, that's to increase the speed and the resolution, the robustness, and potentially quantitation, and also to find new functional contrasts. For paradigm design and signal processing methodology, it's basically towards identifying similarities and differences in subjects, improving robustness, normalizing across subjects and populations, and basically to capture, the goal here is to get rid of the noise as much as possible, identify it, get rid of it, and then capture all the salient neural activity. It's obviously still ongoing. Interpretation of contrast mechanisms in physiology, we really just want to identify the salient temporal and spatial structure of the hemodynamic response. We want to relate this as well as possible to neural activity and also to non-neuronal physiology. There's a lot of physiologic information there that we can look at. And of course, the basic research, we'd love to discover biomarkers. We're still not there yet. Already people like Michael Fox are using it for targets for neuromodulation, which I think is a great potential clinical application. I think it has some real traction. Pre-surgical mapping is already in place. Platform for neurofeedback. Neurofeedback actually has been surprising to me. I never thought it would work as well as it seems to be working. So it might be a real method for therapy. And also, we should never lose sight of the fact that we're actually, our goal is to really understand the brain in the biggest sense. And, a lot of criticisms of fMRI is that it's just cartography. We're mapping the brain. We're mapping different areas as they're modulated with, but we don't really know what's going on as far as that's concerned. And I don't know if fMRI alone will really get us very far in telling us underlying mechanisms. We'll need multimodal integration to really, truly understand the brain. Okay. So the challenges that I'm just going to talk about in my, maybe 35 minutes or so are seamless data encode pooling and sharing, removal of all non-neuronal noise. And these are sort of lofty goals in some sense. I mean, I would love to remove all, I think it's possible to remove all physiologic noise if we want to. I mean, I think that's a long-term goal. Spatial normalization of bold signal weighting across the brain. It's weighted by blood volume in each voxel. We're kind of ignoring that point. Some groups try to get around it. It's becoming more apparent when we're trying to do things like layer fMRI. I'll talk about that. Real-time assessment of ongoing state. So that, in other words, fully understanding resting state to the point where you can put a person in a scanner and read out their ongoing state in real time. And I think that's completely possible. Clinically useful biomarkers. That's, of course, we talked about that already. But the trick there, I mean, it's interesting because the trick there is that so far it takes, many subjects to have a reproducible biomarker. But then the trick that people don't usually talk about is that to actually be a useful biomarker, you have to put a single person in the scanner and say that you're likely part of this group or not, or based on this biomarker. So you still have to have the sensitivity to make a decision about an individual person. It's not just generating the biomarker that's robust. It's actually then using it to say, okay, you have this probability of having this, or we should do this. Other clinical uses, looking at vascular physiology, I think this is a low-hanging fruit that could be one of the first clinical uses of fMRI. There's a lot of practical issues in actually making fMRI clinically useful. There has to be a clinical pipeline, which I think is really, really hard. And I'll mention that briefly. Full individual subject characterization, so similarities and differences. So, right now there's been some work in trying to characterize individual subjects. For instance, I had a postdoc, Emily Finn, who's sort of taking off with this field, sort of looking at cross-subject correlations. And also before that, she did, a brain fingerprint. These are all sort of hints at trying to get, you characterize subjects behaviorally, and you try to find where these dimensions line up with neuroimaging data. And so as opposed to just pooling subjects and looking at trying to find differences, you'd sort of have sort of a method for characterizing their differences and also how they're similar. So I think this is how I think the field will evolve. It has to evolve this way if we're going to make any traction. People are more different than we think, and they differ along a continuum, not discrete differences. So adaptive parcellation. So right now, the way that much of fMRI, especially resting state data analysis, is done is that you parcellate the brain, and there's several parcellations, and then you average all this, put all the subjects in this single parcellation, and then look at the correlation matrix. Well, each subject probably has a unique parcellation, and so if you apply a template parcellation, you're probably mixing more signals than you want, and so that's when I'm talking about adaptive parcellation. I'll even talk about even more of a caveat to that. Layer fMRI, it's one of the things that my group spends a lot of time on. I think it's not only exciting because it's high resolution, but it can open up an entire area of fMRI in which directional, and here I say connectivity, but directional correlations are highlighted, and so you get to understand how brain areas, which direction potentially the communication is going, and also then to pull out the hierarchy based on where the correlation is, depending on what layer it's on. So I think that we need to try to move fMRI more from cartography to mechanistic understanding, and that's usually understanding the underlying scale of what leads to the brain activation as we measure with fMRI. So we need to more and more tie this to models, dynamic models, computational models of brain function. Multimodal integration. Of course, there's many temporal and spatial scales that we don't have access to with fMRI. They're all extremely complementary. The more we can do this seamlessly, the better off we'll be. And determine the correlates. Potentially, I think we have a potential for understanding inhibition and excitation with bold contrast. And I think that one thing that is becoming more clear, at least to me, and maybe I'm late to the game, but most psychiatric disorders are not cortical. Most of them are probably subcortical in nature. And the more robustly we can image subcortical cortical connections and how they vary across subjects, the more traction we'll have in understanding, I believe, psychiatric disorders. And of course, I have a lofty goal of temporal and spatial resolution, 10 milliseconds, 0.1 millimeters, whole brain. I think this is doable. It's definitely doable. It's just a matter of how you want to make your trade-offs. Okay, let's start with the first one. And I'm not going to dwell too long on these. So
Peter, can I interrupt for 1 second just to make sure you're aware of this? We've blocked out an hour and a half for you. So yes, you've used the first half hour, but as far as the class is concerned, we're going to be here till 4 o'clock. So you don't have to rush quite as much as what you may have thought. So you've got another hour to go.
And also, please, if anyone wants to stop me and ask questions, I'm happy to back up and slow down or expound on points as well. So please ask me questions as we go along. Because I'm actually, I don't know how much of this everyone is aware of, or how much experience people have with it, working with fMRI. So if something is just like completely not clear, just let me know, and I'll make it clear. So let's start with seamless data and code pooling and sharing. So this is a problem in the field, and it's been identified many years ago. This was even way back in 1975. There was a task force in NIMH that said, neglected has been the need to bring together and evaluate findings in a given area and consider them in relation to findings from other mental health research, like basically integrating the data. And it's not only data pooling, but it's also just simply having a better model than just having all these papers that are read by siloed groups. The field doesn't progress this way. So it only progresses through synthesis and integration of the results. And that's sort of a fundamental truth, but we don't, when we think of our own research projects, we don't really do that. I mean, to me, the problem comes out even more so. Like, for instance, if I go to the Society for Neuroscience meeting, everyone's overwhelmed. Everyone's, there's so much information. And let's say you might learn like 5 different things and you get excited and you go back to your lab and do them. Does that really, to the extent that we're a network, we're a very unconnected network, is in the sense that there's no body of knowledge that's being added to. It's sort of like very, very fragmented with each individual group and person. So the more we can have something that brings stuff together that we can all look at and add to and understand where the gaps are, I think the field will progress faster. And a brief statement on our publication workflow. So writing papers, having them reviewed seems still outdated. And so basically, the typical publication workflow is that you have the meeting abstracts, paper submission, peer review, and then finally publication. And then if everything goes well, you have clinical trials and implementation clinically. We're not even really there yet with fMRI. We're still sort of in this area right here. And it seems that there has to be a better model for doing this than just through papers in that regard. And so one idea is to, and if certainly papers are written from these, but the idea is that once the papers are written, all the data is shared. And there's there's a growing number of databases. So there's MR specific repositories, OpenFMRI, OpenNeuro. I'm not going to list all of them, but there's a growing number, and they're all extremely useful for. And now one thing I've recognized, even going to OHBM last year, is that I would say that maybe 1/3 of the research is on shared data. 1/3 of these people, I believe, it's hard to get a good estimate, but I got a good sense that so many people have not even touched a scanner. And so they're just analyzing data, which is both good and bad, but I think it's good because I think there's a lot of information to be mined if it's properly curated. You have data agnostic repositories as well. And once again, the big problem is having standards so that this is easily shared. There's NIH funded curated data sets, everything from the Human Connectome Project, which I think has been stunningly successful. I think they should do a second Connectome project, maybe at high resolution or at layer resolution. OpenFMRI and all the way to the UK Biobank, which is incredibly successful as well already. So many databases. I think that they're greatly enhancing the field as far as that's concerned, and changing the way we do science. A lot of us do science. So not only is there data sharing, and this is some examples. There's tool sharing, and of course there's there's standards establishment. So as methods get developed, of course there's platforms. There's AFNI, SPM, and they do get developed and they do get disseminated, which is a good thing. But there's still, the field is advancing extremely rapidly. And so there's certain tools that may not need to be changed as much, like fMRI prep. It's sort of it's a nice pre-processing tool. There's always a tension when you have tools like this, is that there's always going to be potentially better ways of doing things. So you don't want to stifle the field, but you also want to allow it to grow by having similar data types and similar analysis tools. So it's a tricky tension, and I don't think we've completely figured out what the best solution is. Okay, switching gears. So my goal, one of my goals, is to remove all non-neuronal noise. So many papers have been written on this. So what contributes to to spontaneous fluctuations? So I group this into three big areas. One, the good stuff, the spontaneous neural activity, which is either conscious or unconscious or related to arousal and vigilance. This is what we want to fully understand. There's also particularly problematic ones. That's respiration, chest wall, the magnetic field spills over into the brain every single breath you take. But even more problematic is that every single time we take a breath, we take it with a little bit different depth. And that change in depth, the variability of our depth of breathing, varies the CO2 in our blood. And so, and that changes vasodilation at a very slow rate. And that causes a lot of the changes that we see. And we can't, it's funny because we try to use regressors to regress this out, and it sort of defies complete regression. So it's still a little bit of a tricky problem. Cardiac pulsation is a problem. It's less of a problem. It's more well-behaved. Of course, thermal noise, there's even, once you go to higher resolution, your signal to noise goes down such that these respiration problems, all this physiologic noise is less of a problem, and you have more thermal noise. And there's techniques, one called Nordic. It's a nice technique that was introduced about a year ago that seems to reduce thermal noise. And of course, there's motion, which could be considered part of the physiologic, and also scanner instability, which interestingly has become even larger as we push the limits of resolution. There's more instabilities as we push, there's more ghosting, there's different types of ghosting, there's eddy currents, and all these things with scanner instability. It's interesting because the vendors certainly have optimized the scanner for their clinical pulse sequences, but they have, once again, they haven't spent much time chasing down these scanner instabilities that are with these extremely high resolution, high speed sequences that are used selectively for fMRI or even for DTI for that matter. And so this is what the goal is. So this is sort of a classic curve. Many people have done research on it, Larry Wald, and here my group with Jerzy Berduka and Kevin Murphy. And basically the idea is that if you compare the temporal signal to noise, the signal to noise over time versus the image signal to noise, there's a real plateau. The temporal signal noise plateaus at about 100 to 1. And this is all due to physiologic fluctuations, or mostly. So we would like to actually have it such that these curves are, this is aligned with this, so that the goal then is that we can have RF coils that give you image signal to noise of 1,000 to 1. It'd be nice to have 1,000 to 1 image signal, temporal signal to noise to pull out all kinds of subtle things if we just get a handle on our physiologic fluctuations. So that's a challenge. It's worth doing. So what's some of the nature of the of the problem of physiologic noise? So this is what I was talking about. So if you change your respiration over time, this is each breath, and this is the envelope of each breath, and you look at the power spectrum, it lines up a lot with the power spectrum of the fMRI signal. And if you try to regress this out, or if you just take the correlation of this with the time series, you get something like this, which is pretty much, if you even lowered this threshold, it'd be most of gray matter, but it lines up with a lot of networks that we identify as neural networks. So, so that's an issue. and if we try to use this to regress the signal out, it doesn't fully regress out. So here's the global signal. This is actually the voxel-wise correlation with the global signal. If you average up all the voxels and look at the voxel-wise correlation, you get something like this. And certainly that's looks like it's not very interesting. And so people have tried to use that to regress out a lot of this sort of ongoing fluctuations. And if they do that, this is the work by Kevin Murphy, typically what you find is that if you have large enough areas that contribute to the fluctuations that are real neural activity, then what will happen, that will dominate, that will contribute so much to the global signal such that after you regress all that out, then suddenly you have areas that if you didn't do global regression showed no correlation, and in reality they're not correlated, but suddenly they look negatively correlated because you shifted the mean down to zero, down to some lower level, such that areas that were previously, so you can imagine like a boxcar on top of a zero, and then if you take the mean of that whole time series, it shifts down, and then suddenly you have areas that show negative correlation with other areas. And that's what occurs pretty robustly. You get these negative correlated areas that you could misinterpret. It certainly cleans up the signal, but you get that. So that's the global signal. And we're still trying to figure out even really what this is. We still don't know. People think it's just breathing like this. It might be, but it could also be global neural correlations that are some sort of neural activity, maybe arousal or something like that. So it could still be interesting. So the ways of getting rid of the fluctuations that I won't really get into at all, just mentioning that, certainly processing. People use, you can have many different regressors, nuisance regressors. You can apply independent component analysis and choose the components that look the best, or choose the top 5 components, and hope the rest is identify the edges or something like that. People have automated methods for doing this too. There's different paradigms. Certainly cross-subject correlation is beautiful in this regard. Let's say you have a paradigm, a subject's looking at maybe a naturalistic stimuli. Their brain is fluctuating with that stimulus. You have another subject looking at that same time-locked stimulus. Their brains are only going to correlate to the degree that the neural activity is happening. All their physiologic stuff, all their breathing, everything is randomly correlated. So that all gets removed. So that's a really nice, powerful way of getting rid of a lot of the noise. Certainly originated at MGH, actually, with Alex Gamera's using gating. So at the very base of the brain, those structures move a lot. And especially if you're doing, like, for instance, he was doing auditory stimulation. So to make sure that the structure, it's not only the signal change in intensity changing, but it's the structure moving around. If you want to make sure it's in the same spot, you have to gate. And if you gate, because your heart doesn't beat perfectly evenly, you'll have magnetization related to T1 that varies all over the place. So you have to do what's called a T1 correction for that. This isn't very common, but I think for certain things at the base of the brain, it might be important to do. You can certainly have controlled breathing as well. You could have the paradigms in which subjects are trained to breathe in a very regular way. On the acquisition side, I'm sort of a proponent of multi-echo acquisition, allowing you to look at the echo time dependence of the signal. So everything that is bold should show a % signal change that increases with echo time. And everything that's not bold won't. And so it's a really nice way of separating out bold from non-bold fluctuations. And it helps your signal to noise. Certainly navigator pulses usually are used clinically for like gross motion, but especially when you get into multi-shot imaging. So echoplanar imaging, you get everything, you freeze physiologic noise when one signal is shot, where you collect all of what's called your K-space in 30 milliseconds or 20 milliseconds. If you don't do that, if you do multi-shot, then every line of K-space is a different phase of the cardiac cycle, different phase of the respiratory cycle. So that the NMR phase will vary. And so you need navigator pulses that align the phase. That's critical. There's some, company, there was a company, Connecticore, that came out several years ago that allows you to, based on looking at the motion of the head, they had these cameras set up to looking at Morier patterns. They were feeding this into the gradient system and adjusting the gradients to the motion of the head. It kind of works. It's kind of fragile. We have two systems. Neither of them are used that much. I think it might be even, it's more critical, I think, for diffusion imaging and structural imaging. There's a company called Scope that measures magnetic field in real time near the brain. And it might allow you to get rid of a lot of these respiratory changes or things like that. So, and other variations as well. So that's another way of getting at this. But just to reemphasize, the problem's not solved by a long shot. All of these get you pretty close to the solution, but we still have, if you look at any temporal signal to noise, even after all this processing of your fMRI data, at best, it's like 120 to 1. It's not as high as it could be. And so there's still things we're missing. And this is what we're missing, potentially. So this is another approach to increasing the sensitivity, not just averaging the heck out of the data. And also, importantly, not using a single model regressor. So this was a very, very simple task where the subject, this is Javier Gonzalez Casillo, my group did this. Subjects stared at a fixation point. They had a visual checkerboard with either a letter or a number like embedded in it that was hard to see. And they had to press a button whether it was a number or letter. So very simple task. And they did this, they came back for 9 hours of scanning. And so we averaged over 100 runs over 9 hours with a single subject. So it was perfectly registered in space. And so what we found was certainly well-behaved responses, but we found, interestingly, if we did a completely open model, allowing for all different types of responses. And this isn't saying that the hemodynamic response is different. It's the neural activity that it's time locked, but it doesn't behave as you would expect, but it's time locked. So you get almost the whole brain is active. And you start to get certain parts of the brain towards the base of the brain. They have very different responses. They have transients that are negative at the beginning and high at the end that are probably real, that we always miss because, well, one, we don't have enough signal to noise to see them, and two, our models are not adaptable enough to pull them out. Whereas here, it was just relying on the time lock nature of it. So, and you can see only a few of the responses look like this nice standard response, whereas a lot of them are completely all over the place. And it really is ordered. I mean, it's not like it's a salt and pepper. There's an order to the responses as well. Okay, so that's something, that's our goal. I mean, and actually this even tells you as well that in statistical models, when people have a null, they assume like there's a null effect. Well, it's hard to deal with the fact that maybe the whole brain is active and we're just not seeing it. And so it's not a true null in this regard. So how do we deal with that? The other thing that people thought was a big issue early on, it's not that big of an issue with low resolution, but it still is in terms of interpreting the data, is that our bold signal is highly weighted by the blood volume in each voxel. So the venous blood volume in each voxel. So this is a simple experiment where you just, old experiment where I did finger movement, having a subject breathe 12% O2 as opposed to 21% room air. So the arterial blood becomes deoxygenated. And here's where they breathe 5% CO2. So there's a global flow increase. And you see that it's not a uniform signal. You have large vessels and all the veins just popping out as you increase the flow. You have this global increasing flow, a global increase in oxygenation. It just shows the weighting function of the bold response. And So for instance, if you're trying to deeply interpret this data, and I say, oh, well, there's higher intensity, there's stronger bold here than maybe down here. Oops. Does that mean there's more activity? No, it means that likely this is over a large vessel and this is over, more of a capillary where there's only 2% blood volume. So that's something that is not a huge deal because we usually just look at general areas of activation, but it can be. It becomes much more of a big deal when we go to very high resolution. Oh, is there someone in the background?
Could people please mute themselves if you're not muted?
I don't know who. Oh, well.
I've muted them.
Okay, good. That's all right. So, oh, maybe. Anyway, that's all right. Not too distracting. So at very, very high resolution, it's becoming more of an issue because looks like Grant Hartung is, there we go. Anyway, so basically, if you're trying to look at layer specific activation, then if you have heavy weighting of venous blood volume at the surface, it's hard to discriminate upper versus lower levels of activity. So you really need to control for that when you go to this layer level activity. So that's a big issue. Problem is we can calibrate this based on a CO2 stimulus. It's just a matter of, we end up dividing two signals, which throws away signal to noise or reduces our signal to noise. So there should be maybe more robust ways of doing this. All right. Okay, so this is another goal of mine, and I think it's achievable as well, and that is real-time assessment of ongoing mental state or ongoing thoughts, either conscious or subconscious or whatever. Our first preliminary data, this is once again work by Javier Gonzalez-Castillo in my group, in which he had subjects perform these 4 different tasks: rest, 2-back, video, bath, and put the brains in a parcellation and looked at their connectivity matrix with a sliding window and identified 4 categories of connectivity matrices that clearly identified the tasks. So ongoing connectivity during a task, as opposed to just magnitude changes, is very sensitive for identifying what task it is. And it did it with almost 100% accuracy. And then we decided to take a little bit further, and that is we decided to try to, in an unsupervised way, go beyond just simply identifying when and where, what of the 4 tasks are going on. We tried to find, group the time series into sections where there's a uniform matrix. And then what we did was, it's interesting, this is a big problem, is that when you look at a connectivity matrix and you find the areas that are sort of salient or relevant to identifying that task, and you map that connectivity matrix back onto the brain, it doesn't actually look a lot like an activation map. There are connectivities that are outside, that's somewhat overlapping, but it's outside the area of activation that you typically know for like a video or math task, or for instance. So what we have to do then is deconvolve the data in those sections. We have to try to do deconvolution and then compare those maps with neurosynth, for instance, and to say, oh, it's likely reading or likely thinking about memory or something like that. So we tried to do this. It was sort of ambitious, probably too ambitious, but we were able to divide the whole time series into these, probabilities of these various things, periods going on. What I'd like to do is actually create a whole bunch of templates, connectivity templates. You can imagine the brain doesn't have infinite amount of dimensionality in terms of states. You could actually have maybe 50. And then you just compare every TR with a sliding window or some other better method, these templates, and then just read out what a person's thinking, what their state is, and so on, to get some sort of diagnosis. So this was all conscious thought. This is all potentially conscious thought. What's also interesting is that much of the resting state activity is, the one that the resting state that's neuronal, probably a lot of it is not conscious. And Maurizio Corbetta has been working a lot on this. This is some of our own data, but it's sort of along the lines of what he's proposing, is that a lot of it is just simply reflective of how brain areas synchronize during a task. And this was a finding, a preliminary data that we had a long time ago, we never published it, was moving a seed along the primary motor cortex and seeing how well it correlated with the other cortex. And we found a high correlation, as expected, in the motor cortex from the foot all the way to the tongue and everything, but except for the hand area, which was intriguing because, and the potential explanation is that we don't use our hands in sync. We don't actually necessarily, feet you use in sync, legs you use in sync, your mouth and face, it's all left and right is in sync, but hands are not. And so this is reflective potentially of the lack of correlation between left and right hand area. There's still some there. So this is actually potentially very interesting suggestion that this non-conscious connectivity is reflective of basically how synchronized the brain is when it's doing something. Okay, so ongoing more along this lines, actually Laura Lewis, who is at MGH or associated with MGH, sort of pioneered this to some degree, but we've reproduced her results. And we found that, if you take a voxel and you look at the fourth ventricle, which is interesting because it's not in gray matter, and you find that the signal actually correlates with the whole cortex, and it looks something like the Sisbold, but it's actually could also be the fourth ventricle. That when a person is awake, it's higher frequency, low amplitude. Suddenly, when they're asleep, it becomes high amplitude and slow. There's a slow, high amplitude signal. And there's some theories that that's like CSF related to the lymphatic system moving. But regardless, though, it's very robust and it's very much indicative of drowsiness. And if you use that signal and correlate with the rest of the brain, you find this massive anti-correlation in the cortex with that fourth ventricle area that seems to suggest that all the cortex is sort of synchronized not only with this subcortical area, but it's modulated by arousal. So that's another ongoing fluctuation that's potentially very interesting, that's always going on with your data as well. So there's all these embedded fluctuations and changes in states that are going on. Okay, I talked a little bit about this before, but the use of biomarkers. In my process of preparing this talk, I came across a really nice paper that by Parks, and also I believe Danny Bassett's on this as well, basically saying that biomarkers right now, they really aren't working because they lack specificity. That the way we collapse our dimensions of behavior are too simple. There's no specificity to make them powerful. So she or they recommended you adopt transdiagnostic research designs, basically make a wide spectrum of what their diagnoses are, pool the data together based on how they overlap in terms of their dimensions of what their disorders are or not. And then create models that map this full spectrum, and then model the individual's unique functional connectomes. Each one, you have to model that. So that's a tough challenge, but that's sort of the way forward. And this was sort of, and this paper I mentioned, the Merrick et al. Paper, as I read that paper, and I wrote a review, a commentary on it, and my thought was that, of course, the population selection, they were either doing, they're grouping populations based on psychosis versus not psychosis or intelligence. And it's probably too broad. It's probably way too broad. it used one parcellation template. So it used resting state as well, which you can do better than just using resting state. And the one parcellation template certainly, I think probably mixes a lot of the signal from a lot of the subjects. Okay, so the take-home message here is that we need to better characterize individual differences along many different dimensions. So, and that's a challenge, but I think that's it's also exciting because I think the information's there. I think it's a matter of us figuring out how to pull it out. And we also have to better characterize pathologies and compare among and between them. So there might be very low dimensional manifolds of pathologies that we haven't seen yet, that we haven't completely fully appreciated. All right, but there's other low-hanging fruit. It's not just about getting biomarkers with fMRI. I think that if we wait for that, we'll be waiting a long time. There's a lot of really interesting vascular physiology that you can get. And this is two people pioneering this work. There's Ahmed Khalil, who is in Arno Willinger's group in Berlin or in Leipzig. And also Blaise Frederick, actually, his group is looking at this as well. And it's really nice. Basically, the idea is that you take the global signal and you map the bold delay relative to the global signal, the delay, the relative latencies on a voxel-wise basis. And they're able to, this is actually a subject that, or a patient that had a flow deficit, showed up with DWIs, an area of inflammation. And it shows up with, this is a gadolinium study where they inject gadolinium, the standard clinical use right here. But these bold delay maps beautifully show this as well. And so this actually is just as sensitive and doesn't involve gadolinium, could be used immediately clinically, I believe. And this is another technique where Blaise Frederick's group was developing the use of carpet plots. You basically map the time series of every voxel in the brain, and you find, as opposed to looking at the global signal, he finds that, typically what happens is that you have these ripples. Subject takes a breath, or they move a little bit, or they do something, or not motion, that would be a different sort of ripple, but there's some sort of global change, if they sigh or something like that, that changes the oxygenation. If you reorder the time series, you can actually pretty nicely get a sense of the transit time through the brain. And that's very clinically relevant of getting transit time based on the delays and how they order with bold contrast in the brain. So here we mapped out a delay map with gadolinium, delay map with just subjects lying in their scanner doing nothing, and was able to get a nice carpet plot that transit times of -4 to 10 seconds, or actually 4-second transit time. So that's an easy clinical application. Okay, so what about a practical clinical pipeline? So MRI is so incredibly successful because you can do a scan and you can put a subject in the scanner and immediately out pops something like this, whereas either with a diffusion weighting scan or T1 weighting, you get some clear identification that a radiologist can just look at and say, oh, there's something obviously going on here. With functional MRI, it's different. So here's, even with a PET scan, it's only a single metric, like usually it's metabolic rate or something like that. With structural MRI, you clearly see, it's just maybe a couple of parameters, T1 or T2, you have different contrasts, you see what you see. With functional MRI, the challenge is huge. You have literally hundreds of ways of making this map based on all the processing that you're learning in this course. And all the processing that I'm mentioning today is that there's many ways of making this map, and there's many ways of making it wrong. And each step somehow might change the results, which may or may not be clinically relevant. So right now, it's so difficult to come up with a standard processing pipeline because there is no necessarily best pipeline that that people agree upon it's like the most robust and the most sensitive, or hits the sweet spot. And in subjects, if they move, how do you deal with that? It's more of an effect of motion than with other scans. So it's extremely difficult because there's so many different ways of making these maps. So you need to make it reproducible, robust to motion. It has to be standard, simple, and FDA approved, which is another hurdle. It needs to be updatable as technology gets better. Certainly that's the case with most MRI scans, and easily disseminated. How do you do that well? So it's a very hard problem. That's why that's why the vendors aren't really embracing it. There might be third parties sort of starting to embrace clinical packages, but it's truly challenging for this reason, that there's so many ways of making a map and so many ways of making it wrong. All right. So full individual subject characterization. I sort of dwelled on this a little bit, probably enough already, but individual subjects. I mean, I think that a lot of fMRI in the past has been trying to get rid of this, just average them together and hope to see an effect. And that works. That's great for looking at group differences in some way, but I think that we're finding that the problem is much more complicated than that. And there's many different types of individual differences. There's, structural differences. There's resting state or individual differences with specific naturalistic tasks. So one clever thing that's being done with naturalistic tasks is using it as sort of a litmus test. It's sort of finding the right task that optimally pulls out differences. Like, for instance, if you have a tendency for paranoia, for instance, you might, if you're listening to a story or seeing a video that is ambiguous in some way, your differences might be pulled out more than other subjects. So once again, Emily Finn is trying to develop these sort of paradigms. And I think this sort of thing to identify these traits, I think is promising. The whole idea of having like a fingerprint or identifiability of the individual subject, being able to separate them from the rest and then make a statement about them, I think is powerful. And other parameters you can measure with fMRI, it's not just resting state connectivity, it's cross-subject connectivity, cross-subject correlation, the states, like if you do a sliding window, their connectivity states, how long they stay in that state, how they transition, maybe the sequence of transitions of states, transience of the bold signal, things like that. There's many parameters to measure that we really haven't delved into in the field that we could pull out. All right, this is the problem with parcellation. So this is just one example that was brought up in a paper that's mentioned down here. And of course, everyone, if you want, obviously Bob will share this talk, but I mean, you can have these slides, you don't really have to take notes. I can send you them or whatever. So with brain parcellation, there's one study that said, well, there's all these assumptions that that parcellation, the way you parcellate the brain is the same from children to adults, which is probably not the case. The network properties are the same. And the parcellation selection doesn't really have an impact on results. I think that's all of these things are probably not true. I think that if we parcellate the brain, we think we get a handle on it, but in fact, we're potentially introducing more variance by how we parcellate. And the reason why I say global hyperalignment, I don't even mention Jim Haxby's work, but his idea is that you align, rather than aligning based on structure, you align based on function. So you find the functional areas and then you align to that as opposed to purely the structure, which there are differences. So there's many different atlases. There's many different ways of parcellating the brain. They all, as you can see just by casually glancing at this, they're all kind of different and they all have different boundaries, and that all makes a difference. So I would say that another big challenge of the field is finding a way to parcellate individual brains in such a way that then you can average them together or use them in some way that's a little bit more robust. And here's another potential caveat as well, that it's more of a data that based on that's backing up what I just said in the previous slide. This is some work by Prantik Kundu when he was in my group, using multi-echo acquisition. It turns out that if we do independent component analysis with multi-echo, we can reorder the components such that there's a natural cutting off of where this curve kind of reaches into the noise. And it's a nice way of selecting components, which are aligned very much with the parcels. You almost use them interchangeably. And it turns out that what he consistently found is that in young subjects, there's many more components than with older subjects. And so just plotting this curve, you have subject age versus the number of bold components, or essentially parcels that might come out. And it's always higher with younger subjects, and it sort of settles in by about age 40. So that's an issue. And in a sense, this is sort of like a biomarker in itself, that you have the parcels change. They change in size, they change in number. And it's not to say that the actual functional area of the brain is somehow changing significantly, but it might be more for younger subjects, it just might be more divided, and it might settle into one sort of group sort of connectivity within that parcel as you get older. But that's a factor. Okay, I'm going to talk about just layer fMRI quickly. So layer fMRI, for those of you who don't know, is basically a way of sort of bridging this gap between these very invasive approaches of looking at individual neurons or groups of neurons with calcium imaging to typical fMRI that looks at either blobs, correlates that with behavior, or looks within areas. So it turns out that one of the more interesting scales of brain function is at the columnar and layer level. There's a lot of interesting cortical circuitry that happens at this scale. And what's also useful about layer fMRI is that it could cover the whole brain. So it not only has this resolution that bridges these methods, but it has the coverage of the whole brain, which could advance in terms of how we understand how areas that are far apart are communicating. Okay, what's needed for layer fMRI? I'm not going to go into too much detail, but basically you need voxels that are below 0.75 millimeters, achieved through multidimensional acceleration or parallel imaging. High sensitivity, you need at least, you can do it at 3T, but 7 Tesla is the best way to go about doing this. You need high specificity. What I mean by that is you need pulse sequences that look at more capillary effects as opposed to the large vessels. And you need special processing methods. And the basic idea, and this is a huge oversimplification, is that our brains are on many levels prediction engines, and all the way up. And so you might have, for instance, V1 gets input, and that you might have, it feeds into higher brain areas from upper layers into the layer 4. Everything that is higher gets fed into by lower areas through their middle layer, and everything that is fed back, these higher brain areas that form models of the world or predictions, then feed back to modulate activity in V1. So, and where they feed back is in upper and lower layers. So if you see activation or connectivity or correlations in these upper and lower layers, you can imagine that it's a activation that is dominated by feedback. If you see activation like across layers, that this is from the CSF down to white matter, that shows this sort of thing, you can imagine it's receiving feed forward from a lower area. And these are the canonical sort of shapes of connectivity profiles that we found. And I'm not going to go into the pulse sequence. Basically, it's our primary sequence that we use is called VASO, and that selectively images blood volume, which we were surprised. I was very surprised to find that the blood volume changes in the brain are much more microvascular than, for instance, fold changes, and it's sort of on the line of perfusion changes, which, you know, if you've listened to, like, if you followed the history of modeling fMRI, there's the balloon model that sort of suggests that you have these large veins that sort of expand. That's probably not happening. It's probably capillaries that are being recruited, or microvessels that are increasing in diameter. So that's a nice thing. And the reason why is because if you go down to about 0.75 millimeter, you get something that goes from a blob down to what looks like two very discrete lines. And this is looking at motor cortex just with simple finger tapping. And we've been able to map this sort of activity and connectivity. This is just an example. And you'll be hearing more about, you know, seed voxel approaches, maybe you already have, but if you just look at the data and you take a seed in one area, in this case, you take a seed in the upper layers of M1. So that's a seed right here, this teal color, and see what's correlated with that. You find that all of sensory cortex seems mostly correlated with that upper layer, which is expected because it receives cortex and thalamic input. If you switch the seed, if you just shift that seed just a little bit along the layer, you get a completely different area that's correlated. This is more prefrontal, which could be both, well, in this case, that is suggesting that it's output, but also it could be feedback from these higher areas, the premotor cortex as well. So bottom line, you shift the seed a little bit along the voxel, you get very different areas. And you could do the same thing and shift, instead of having the seeds along the layers, you could have the seeds like in the areas, like seed in S1, looking at motor cortex. And then if you look at the signal in the layers of motor cortex, you find what's expected, the cortical inputs right here. That shows the highest power of correlation. But if you put a seed in premotor, you find this double peak, which suggests that the premotor is sort of feeding back to motor cortex right here. So I have to remind you that this is the same region of interest. This is the same layer that we're looking at. We're just taking different seeds on where and then seeing the correlation with different seeds. So that's kind of exciting. That's an example of layer fMRI. Without going into too much detail here, we did the same thing with V1 and looking at how it relates to taking seeds in MT right here, and also the lateral geniculate. So the V1 is, sits between these. It's higher than LGN and it's lower than MT. And so the idea is that you would have, as it turns out the case, if you just look at the V1 layer profile right here and take a seed in LGN, you find that the layer profile looks something like this, which suggests that it's feeding in, it's feed forward from LGN. Whereas if you look at a seed from MT, you get this double hump right here, which suggests that it's feedback from MT. So once again, same region of interest, just different seeds. And we expanded on this a little bit more, and we found that if we take the seed and just move it along, so here's starting with the seed in the thalamus, and thalamus basically feeds into the entire visual cortex. But if you move that seed along the cortical ribbon, you find that everything below that seed, in some sense, hierarchically, now has a feedback profile rather than the feed forward profile. And that keeps on shifting as you go along the cortical ribbon all the way up to MT. So that's another way of getting hierarchy. And we're just starting that. So I've showed this results because we're still trying to figure out how to do that robustly in the whole brain. And here's the problem. So we can image whole brain with layer fMRI resolution. And let's say if we start out by making a connectivity matrix at high resolution with each maybe a parcellated each cortical region, you have a lot of parcels. And so it's already a large connectivity matrix. But then suddenly, if you then take each one of these parcels and divide it into now layers, then the whole matrix explodes. And so it's very, very hard to really get one's head around the nature of how one layer speaks to another layer within and across all the parcels in the brain. It's an incredibly multidimensional problem that we haven't quite mastered yet. Take a lot of computational power, but there's different ways of taking stabs at the data. Like I said, like this way is one. It's more of a hypothesis-driven sort of approach. And so, yeah, we can make these connectivity matrices across layers within these networks, which works pretty well, but it still only gets a fraction of the data that we collect. So it's a real challenge to actually not only get robust, high sensitivity, high resolution images at this resolution, and to select just the vasculature that's sensitive to neural activity, but it's just hard to really make sense of the data. It's computationally currently kind of intractable to do it completely. Once again, we tried doing more hypothesis-driven approaches where we've looked at, for instance, V4. We take a region, we look across the layers in V4, and we take all these different seeds in areas that are below and above V4, and we find, as expected, sort of areas that the lower areas, all the visual areas, feed into V4. They show the V4 shows this single hump, and all the areas. If you're looking at V4, you're looking at, if you take seeds from areas that are above V4 that feed into V4, feed back to V4, you find this double hump for the most part. And it's not perfect data. It's very preliminary. So, all right, just about getting there. So migration from cartography to mechanistic understanding. So right now we're limited to just this spatial scale in the brain. So we're limited to millimeters, down to millimeters, and maybe seconds or a little bit faster than that. So, and it's an inverse problem, essentially. There's many different mechanisms that could explain our maps and explain along those time series. And to get at those mechanisms, we need to have other measures and models that we build and iterate off of to get a deeper understanding to do this. So that's all I really have to say about this, other than it is, these are the sort of things that sometimes keep me up at night, thinking, are all we doing is just cartography with fMRI? We're just mapping. And that's not really understanding, just mapping. And so we need modeling. We need modeling to make sense of these maps and to add depth and insight based on other modalities. So, and then a really beautiful paper, not going to go into it at all, by who I really respect in the field, Pascal Fries, just talking about how you really do, we do need to find better ways of integrating across spatial and temporal scale to build models that lead to understanding the brain, to true understanding. So this is sort of where we're at. We're good at mapping, we're good at collecting this data, and we need to start spending more time building better models that we can iterate and build and understand. So just another quick example of, this isn't necessarily related to what I just said, but a beautiful example, one of my favorite papers at the time of doing multimodal imaging is looking at monkeys and doing electrophysiology on monkeys. Basically, this is when monkeys were sleeping. And while they were sleeping, their hippocampus has these ripples that are triggered that you can only measure and detect with electrophysiology in the hippocampus. So every single time a ripple was triggered, basically it was marked, and hemodynamic response was convolved with that. And he found that these ripples actually communicate, or there's correlations with the rest of the cortex. It basically suppresses subcortical activity and shows strong activity throughout the cortex. And certainly in the paper, he talks about mechanisms for memory consolidation and things like that during sleep. But it's a beautiful example of just using simple electrophysiology as a temporal timestamp of something that he knows is relevant to memory, potentially, and showing how, by convolving with the hemodynamic response, how it communicates with the rest of the cortex. So, nice example. Determine the neural correlates of fMRI. I'm not going to dwell on this much, but basically, there's some studies that have looked at applying, for instance, muscimol, which is a GABA agonist, which increases inhibitory neural activity. And if they do that, they actually see a huge modulation in the bold response. You have much more of a difference between the initial transient and this steady state, and maybe a larger post-undershoot. And Sanji Kim's group just has a paper out on using optogenetics to selectively excite inhibitory neurons or excitatory neurons. And what's interesting is that if he selectively excites inhibitory neurons at different frequencies, he gets very different responses. Here's at 1 hertz, and you get this, what's typical, looks like typical bold, transient, and then some steady state. If he stimulates inhibitory neurons faster, you get a transient, and then a huge undershoot right here that dominates. So this might be suggestive that these all these transients in the bold response are very much of an interplay between excitation and inhibition that we just need to model to pull out a little bit better. Okay, subcortical connectivity. As I mentioned, I think that the next frontier in neuroimaging in terms of understanding psychiatric disorders is really understanding not only with diffusion imaging, which does it spectacularly well, but with functional MRI, as to get at this subcortical activity. And this is the results that I showed you from Javier's 100-run averaging. And you get this nice structure of clear differences in activation that don't look anything like the canonical response in the subcortex that show activation in the thalamus, actually, that show activation with this task, that we just need to find better ways of image at high resolution with more robust emotion with subcortical activity. Okay, and there's another nice, I just found this paper just recently, a really nice paper that shows connectivity matrices with cortex and also the covariance matrix with various areas in subcortical areas. Okay, I think we're getting towards the end here. So how do we get this high spatial and temporal resolution? So, so I think that we can certainly get to a spatial resolution almost as high as we want if we're willing to sacrifice the time. And if we time our data to, like, for instance, a stimulus, we can chop up K-space any way we want. So really nice work actually at MGH with actually Cowan Setzenpop and his graduate student looking at EPTI, which is a way of chopping up K-space in arbitrary ways to get at either extremely high resolution or at getting at other information. The problem is that it takes time and it increases variation, increases the temporal variation. So we need better ways of correcting for these misalignments in K-space when we chop it up like this. Certainly going to high field, I think 70 is enough to get to this resolution. Local head gradient coils give us a little bit more efficiency in time. Precise acquisition and stimulus temporal resolution. That what I mean by that, and I'm going to get to that in the last thing that I'm going to show, and that is if you segment your K-space, you can actually time it such that you bin your K-space data based on when the task is going on. So you can measure your task and then keep on just collecting your K-space data and then bin after the fact to get nice images of pre-stimulus, post-stimulus, and whatever. So precise timing is necessary. We have most of that. We can do most of that. Certainly local RF coils local shim coils would be important as well. We can, I think, once again, MGH is pioneering using the RF coils as shim coils. That's a clever idea. That's, I think, all of these methods are amazing. It's just that they're not disseminated because we rely so much on the vendors. Until there's companies that disseminate this, it stays within, Larry Wald's group, or and so we really do need to get clinical applications to get the vendors involved so that we can disseminate all this cutting edge technology that's all over the world. I think there's still some mysteries with microvessel sensitivity, and certainly John Polomini is modeling this as well. We're still getting an understanding of this. And certainly parallel imaging is great, but we've even found that when we try to do layer fMRI, if we push things really hard, there's eddy currents all over the place that cause artifacts that we still need to correct. And so it's still an ongoing struggle. Okay, the very last thing. I wanted to show this because this is sort of in line with potential controversy or a challenge. It's always been a challenge in fMRI to look at neural activity directly. My own group has tried to look at neural currents. Theoretically, if you have a current through dendrites, it should cause a phase shift. Turns out it's very, very hard, if not impossible, to see in people. This paper came out last week, and it's one of the most, if not the most convincing paper that I've seen. And it's not looking at neural current effects. It's actually looking at what they claim is a change in T2, pure T2, not bold related T2, due to the polarization of each membrane. And that changes the configuration of the membrane. Either the hydration slightly swells slightly, or there's some other configurational changes that change T2. And what they found, they're at 9.4 Tesla. And this is the reason why I sort of dwelled on this case-based chopping up approach, because they used this. They actually, they collected 5 milliseconds, they had a TR of 5 milliseconds, a TE of 2.5 milliseconds. And they collected one line of case space at a time, and they timed it to the stimulus. So the stimulus was timed, and so you basically collect images across time and then across case space. So each image would be like the first image would be the first 5 milliseconds. Eventually you would build it up. It'd be the first 5 milliseconds, then the next 5 milliseconds, and so on. So they made time series of these. And what they found is that they did basically electrical stimulation of the whisker pad. And they found that within 20 milliseconds, they saw, or 15 milliseconds, they saw a pretty robust response. It's only 0.1%, but it's very robust. And it seems to correlate in latency more to spiking. They also did simultaneous electrophysiology and local field potentials. It's in line with spiking. It's a little bit before the local field potentials. And once again, it's very robust. And they did something clever. They did optogenetic experiment where they first did the whisker barrel electrical stimulation. They saw thalamus activation first, and then the contralateral cortex. But then they switched the order and did optogenetic experiment stimulation of the cortex, and then that would cause a cascade from cortex to thalamus, and they saw the order reversed in their measurements. So that was pretty convincing. They did some control experiments for proving it's not bold. And one thing interesting is that they actually they did an experiment where they looked at T cells and changed the polarity just by adding different amounts of potassium, and found the T2 varied by up to 20 milliseconds from 0 to 150 millimoles, just and corresponding to the polarity of the membrane. And so in doing this, they were actually able to show that, this is my last slide on this, but they're actually able to show that even at the layer fMRI level, that there was hyperpolarization that occurs initially that seems to suggest a wave of inhibition that's acting on the area, and then depolarization that occurs a few milliseconds later in other layers. They were showing within layer or cross layer propagation of the signal. So honestly, I don't know completely what to make of this yet. It's super exciting. It's believable. They have a suggestion that it might be doable. I mean, this is at 9.4 Tesla, at 7 Tesla with humans. Neural density, it turns out, is less in humans, and so the signal will go down. But if you have larger voxels, then it would go back up. So it might be, you're looking for a 0.1% signal change. People move, timing isn't precise. It's really hard. You have to divide up case space like this and then correct for it, which is also challenging. People will find it more and more, I think. I think it actually is a real effect. And this could be this is a huge challenge, but it could be the future of some aspects of how we do fMRI. So last slide, still the big challenges. I think we should never lose sight of the fact that what we really want to do is one of two things, or both. We want to truly understand the brain, not just map it and say interesting things about what's happening where. We want to understand how it's organized. We want to understand how it works, all the mechanisms. So fMRI certainly will help, but it won't answer all those questions, of course. But to the extent that we can add models to fMRI and make sense of fMRI data with models is a good thing. We'll push the field forward. And we also want to have it useful clinically. I think it can be if we get it more of a handle on individual differences. And there'll be a lot of work with that, I think, in the future as well. So that's, I think, still our big challenges. And I'd just like to thank my group, all my staff scientists, all the postdocs and students and postbac artists, and also visiting professors. And with that, I'd just like to end. Thank you.